All right, I'm going to back up a little bit from where I was last time. There we go. Had to re-put my password into the iPad. They make you change your password every 6 months. Meanwhile, my Auburn Lee County campus password's the same one that I started here with in 2012, and amazingly no one's hacked it. Just saying. So, all right. I'm going to back up a little bit because we're going to talk about the mechanics of replication, but I want to show you guys again in the prokaryote model of replication where it's a circle, and in the eukaryote where it's linear, it's the same thing here. So you get replication starting, and then you get an opening of the double-stranded DNA to form single-stranded DNA, and then new DNA is put in there. In both prokaryote and eukaryote, you have this replication bubble that expands because at each end of the bubble there is a replication fork, one there and one there, and they are moving away from each other because more DNA is opening up to be single-stranded, copied new DNA is put on there, and once the DNA is opened up, it stays open with the new DNA being put on there. All right. And in eukaryotes, it's the exact same thing, only because you have multiple origins of replication, you get multiple replication bubbles, but still each replication bubble has a replication fork at either end, and they are moving away from one another. It's just that in prokaryotes, the replication forks move around until they meet, and that's the end because there's only one origin of replication. Then the whole thing is replicated, and the two newly replicated DNA molecules have to be cut apart by topoisomerase II. Here, when two replication forks meet, or two replication bubbles meet, the bubble pops. So this is all one piece of DNA here, one strand, and this is all one strand here. This is going to open up, this is going to open up until they meet, and you get this, right? And we talked about the mechanism by which new nucleotides are added. So nucleotides come in, and the reason why this phosphate gets close enough to be attacked by that hydroxyl is due to the base pairing. So a G means that only a C, except for rare cases where a mistake is made, only a C will fit in tightly enough so that this alpha phosphate, or the first phosphate, outside the 5' carbon is close enough to be attacked by the 3' hydroxyl group. This releases the beta and gamma phosphates, so alpha, beta, gamma, that's just how they're listed. This is called a pyrophosphate. It comes off, releasing a little bit of energy, and then the pyrophosphate spontaneously breaks apart into two inorganic phosphates, and that releases a whole lot of energy, and that's what drives this reaction. So the enzyme directly doesn't require energy to work like ATP, but ATP is actually being spent on this because the chain of events or chemical reactions in which the phosphates are added do require ATP, and so that's where energy is added so that it can be released here. And again, the polymerase is dumb. It does not know, it does not read the base sequence and know which nucleotides to add. Instead, like I said, it's whichever one can get close enough due to the base pairing. All right. So I wanted to go back and show you the replication bubble in prokaryotes and eukaryotes because for the next few slides we're going to be looking at a replication fork. But remember, this replication fork is only one of two forks in the bubble. So there's going to be another one over here where I'm standing to the left that is the exact same thing. This replication fork is moving from left to right because that's the way the DNA is going to be unwound. The other one is over here by me, moving from right toward the left. We'll go back and put the bubble together again in just a little bit. So, look at what's printed at the top of this slide. I want you to memorize this. Burn it into your eyeballs, right? So when you close your eyes, on the back of your eyelids, it says, DNA synthesis always takes place in a 5' to 3' direction. Let's go back and look. See 5' up here, 3'. Synthesis takes place in a 5' to 3' direction with respect to the DNA being synthesized. So the 3' end is the end of the chain, and new molecules are being added at the 3' end. Reaction occurs with the 5' end of the nucleotide, but they're added to the 3' end of the chain. You'll note because of the anti-parallel or opposite orientation of the strands in a DNA molecule, that the new strand being synthesized in a 5' to 3' direction is moving down the template strand or the existing strand in a 3' to 5', the opposite. So you have to remember that DNA synthesis takes place in 5' to 3' direction, and the most important thing is that it's with respect to the strand being synthesized. Because if you just remember 5' to 3', you're like, okay, is it 5' to 3' with respect to this new strand or the template? All right. So the existing DNA is being unwound over there, leaving two strands, the darker gray and the lighter, well, this one on top and this one on the bottom to be copied. Because DNA synthesis always takes place in a 5' to 3' direction with respect to the strand being synthesized, you can see that as the DNA unwinds, this strand can just have continuous synthesis here, right? As more and more DNA opens up, it can just keep going. We got a problem with this one up here, however, because you're going away from, synthesis has to take place away from the replication fork. So you can do synthesis this way, but eventually you're going to hit something and have to stop. Now you're going to have to wait until more template opens up and start somewhere over here and make another piece, and then the polymerase is going to hit that 5' end and stop. So we'll look at this in more detail coming up, but you can see that one strand is going to have continuous synthesis. That's called the leading strand. And the other strand is going to have discontinuous synthesis. You have to make one little piece, then make another little piece, and then make another little piece. And the reason for that is because of the direction you're going. Right? This is called the lagging strand because synthesis here lags behind the leading strand. And that makes sense because the leading strand is going as fast as the DNA is unwinding. The lagging strand has to wait until enough DNA, usually about 200 bases or so, opens up, synthesize a piece on that, and then it has to wait till another 200 bases of template DNA opens up. And it has to do this repeatedly. All right, so as the replication fork moves from left to right, more DNA is synthesized, but you can see that on the leading strand you get synthesis here on the newly opened up. Here, not so much. You have to wait until enough opens up from here at the top to the middle panel where you can start again, make another piece. The leading strand is continuing to go though. Then the lagging strand up here has to wait until another little bit opens up, and then you can make another piece. See how that works? And so you have continuous on the leading strand, discontinuous on the lagging strand, and these little fragments are called Okazaki fragments because of the person who discovered how all this works. And they're going to be okay. We'll see when we talk about the proteins that take care of DNA synthesis and fixing up after it's over. We're just going to have to join these little pieces here. So you have complete base pairing here. You have complete base pairing here, here, and here on the Okazaki fragments. The only thing that's right here in these little gaps is there's no sugar phosphate bond there. There's not a space where there's a nucleotide on the template strand that doesn't have another corresponding nucleotide on the newly copied strand. It's just that the only way polymerases can make phosphodiester bonds is by adding new nucleotides to the chain. So when the last nucleotide is added here, the 3' end of the last nucleotide is just hanging out. The 5' triphosphate here is still there on the first nucleotide of the previous Okazaki fragment, but the polymerase can't join that because that's not the reaction that it catalyzes. We're going to need another enzyme to come in and help us at the end here. All right, so here's the theta replication model that I went over at the beginning again, and this, from where I'm drawing the line to the right, this is the replication fork that we've been looking at. But I said keep in mind that there are 2 replication forks in every replication bubble, and here, from where I'm drawing this line to the left, is the other one. This can be slightly confusing if you don't think about it the right way, but look at the replication fork heading to the right. You can see that the leading strand is on the bottom here, and the lagging strand is on the top because the leading strand is able to keep going 5' to 3' as that fork opens up. But if you look at the left fork, the leading and lagging strands appear on opposite template strands of the template DNA. Whereas over here on the right side, the leading strand is on the bottom and the lagging strand is on the top. Over here on the left, the leading strand is on the top and the lagging is on the bottom. Why is that? Because the left fork is moving in the opposite direction. And so on the left fork, it's this strand that's being synthesized 5' to 3' in the direction that the fork is opening up. So which one's going to be the leading and which one's going to be the lagging strand? The leading strand is going to be the strand that can be synthesized 5' to 3' in the same direction that the replication fork is opening up, right? So you have to look at the polarity of the template strands to figure out what the polarity, which is going to be opposite, of the newly synthesized DNA strands is going to be, and then figure out which one is moving in the same direction that the replication fork is opening up. Okay? Everybody's too quiet. Too quiet. This is the less asking, or this is the less I do problems with you part of the, part of the course where we're just learning how these processes work. But it's still really important if you are not sure, if you are at all unsure about how something works, I guarantee there's multiple other people who have the same question, but I can't guess what it is. So put your hand up and ask, and we'll talk through it. All right. So, yeah.
I don't get really what's going on here. How the lagging strand is right behind the leading. I just don't see it at all.
All right. So this is. I wish that this was rotated like this, but if you have this theta replication is rotated on the top, it's going to look exactly like this. Let's look at this one. All right. So look at the ends here, right? So you can see which end of the template strand is 5' to 3'. Let's pretend that these, and I'll go through and do this again in a minute. Let's pretend that these newly synthesized strands are not there at all. So don't look at those, even though their answer is right in front of you. So look, here's 5' and here's 3 prime. So on this bottom strand, if the old strand is going 5 prime to 3 prime right to left, the other strand, the one we're making, has to go 5 prime in the opposite direction, so left to right. Does that make sense? All right, we're good? So, and then on this one, it's going to be the opposite. It's 5 prime to 3 prime left to right on the template, so that means the newly synthesized strand is going to be going in the opposite direction, right to left. So we got that. If you look at both the leading strand and the lagging strand, the arrows showing which way replication is happening are both going the same direction. That is because DNA replication only takes place in a 5 prime to 3 prime direction with respect to the strand being synthesized. So let's cut it in half at the origin of replication and say, is this going to be the leading strand or the lagging strand? Pretend you don't know the answer. Synthesis on this strand has to take place from left to right, and this replication fork is also unwinding from left to right. So you can see that synthesis is going this way, and it can keep going continuously as more and more template DNA gets unwound, right? But on the other strand up here, because this synthesis is going right to left, you can make a piece that goes this way, but it's going to hit that end and stop. Then you have to wait until a little bit more DNA unwinds and make another piece, another Okazaki fragment. Then more has to unwind. Let's look right here at the fork itself. This fork is unwinding from left to right, and as it does, more new nucleotides can be added directly onto the leading strand, but no nucleotides can be added onto here. Why? Because it's a 5 prime end, and that's not the way the chemistry works. So on the lagging strand here, you have to wait till probably this much, way over to here, this much template DNA is going to have to open up, and then you can start here and go that way until you meet this end. Right? Does that make sense? Is that helpful? All right. Yeah. So both the leading strand and the lagging strand are synthesized 5 prime to 3 prime. That's the way it has to go. The leading strand, here, the leading strand 3 prime end is always going to be pointing toward the way the replication fork is unwinding. The lagging strand 3 prime end, here, here, and here, are always going to be pointing away. So yes, you're right. The 5 prime end of the 5 prime end of each Okazaki fragment on the lagging strand is going to be closer to the replication fork than the 3 prime end. So usually this, like that, is okay, but then somebody would be looking at this end and they're like, well, this is the leading strand, so this has got to be the leading strand too. But it's not the case. You have to consider each half of the replication bubble separately, because this fork, here's where it started. So it's starting here and going to the right, starting here and going to the left. It's going to be the opposite for each of them. So going to the left over here, the strand that was the leading strand on the rightward moving fork ends up being the lagging strand, because both strands are pointed in the same orientation. They have to be, because the DNA has to be in anti-parallel form, meaning one strand's 5 prime to 3 prime left to right, the other one is 5 prime 3 prime right to left. Did that a little bit? Still good? All right. Then over here now, we're moving this way, so this strand is going to be the leading strand because its 3 prime end is pointed the same way that the replication fork is unwinding. So as soon as more template DNA unwinds over here on the left, you can immediately keep adding more nucleotides going that way. But just like this strand had to wait until this top strand on the replication fork opens up on the right, this lagging strand on the bottom left has to wait until more of this opens up because it has to be synthesized right to left. So when in the same replication bubble, on both sides of the origin, the 5' to 3', 5' to 3', 5' to 3' polarity is the same on both sides of the origin of replication. However, which is the leading strand and which is the lagging strand changes because of the direction the replication fork is moving. So DNA on the bottom strand, it has to get polymerized left to right because of the chemistry. It's just that this one is moving to the right, this fork on the right is moving to the right, and the one on the left is moving to the left, so it's the opposite. Make sense? So the way to figure this out is that you look at the template to see which way the newly synthesized DNA has to be moving or being synthesized. And then you ask, is that the same direction that the fork is being opened? If the answer is yes, then it can keep going continuously and it's the leading strand. If not, then it can't, it's the lagging strand. Good? All right, so let's talk about all the proteins that are involved here. Screen's pretty close there. All right, so the first thing we're going to talk about are initiator proteins. So initiator proteins make a complex that interacts with sequence at the origin of replication and causes the DNA to deform such that some of the double-stranded DNA opens up. Once you get that unwound a little bit, then you add our favorite protein, the DNA helicase, right? That's this guy right here and that guy right there. DNA helicases are hexameric proteins, which means there's 6 subunits that form in a circle with a hole in the middle like a donut. And the initiator proteins help to add the helicase to each of these strands. And they do it like this. So if this is the circle of the 6 helicase proteins, they open it up and they close it around the single strand of DNA. And so the helicase is hooked onto the DNA very tightly because it's a circle around the DNA that can't come open. The helicase burns ATP for energy and pushes against the double-stranded DNA, busting apart the hydrogen bonds that hold the DNA strands together. And there's one of these at each end of the fork. These are the proteins throughout replication that are responsible for unwinding the DNA. As soon as the DNA is unwound, you can see these little purple guys here coming in. They are called single-strand DNA binding proteins. They will bind to the DNA and they perform 2 functions. The first is they prevent the DNA from coming back together. So this DNA was together due to base pairing. If the single-strand DNA binding proteins are not there, it will snap right back together. Right? And so it keeps it separate. Also, remember we talked in DNA structure about the double helix of DNA protecting the groups of protecting the side groups on the bases from interaction with harmful free radicals and other bad molecules that are hanging around in the cell. Once this DNA versus RNA, once this DNA is opened up, you got to protect it from that as well, protecting it from damage. So the single-strand DNA binding proteins do that too. All right. In front of where the DNA double helix is opening up, proteins called DNA gyrases are present. DNA gyrase is a topoisomerase. Does anybody remember? Well, it says what that is. Topoisomerases are capable of breaking the covalent bonds of the sugar phosphate backbone and allowing rotation and then resetting those bonds. Every 10.5 bases or so in our DNA that gets unwound is taking one full turn out of the DNA. Remember, it's one turn for 10.5 bases in B form DNA. B form is really 10, but it's kind of like 10.5 in live cells. So every 10 bases, you're adding a turn or removing a turn. What happens when you remove turns from the DNA or add turns? What happened with the piece of rubber hose that I had up here when I added or removed turns? You add supercoiling because the DNA wants to come back to the way that it is. So whether you have a circular DNA molecule in prokaryotes or a linear DNA molecule in eukaryotes, all this unwinding is going to make supercoiling of DNA over here and over here. And if that supercoiling doesn't get removed, it's going to get to the point where we're in trouble. So I gave one example when I was talking about supercoiling, and that was the telephone cord. I'm going to pick on the ladies now. If you have a curling iron, right, or a flat iron, think about, so what exactly right now does the cord to your appliance look like after a long time of use? It's a jumbled mess because you're turning it and flipping it around as you're using it, and I wish these guys get here at 6 a.m. and I wish they would do the blowering then and not now. Anyway, the supercoil, the cord's in a big mess, and can you stretch it out the whole way? No. And so the same thing will happen here and here. If the DNA gyrase enzyme was not able to relieve that supercoiling, DNA replication would stop dead in its tracks because the DNA on either side of the fork at either end of the replication bubble would become so jumbled up that it would not be able to unwind any further. So here's the replication bubble. It's got a fork at each end, and the forks are moving away from each other, right? So there is one DNA gyrase at each end of the replication bubble, right? You can say that it's in front of each fork, or you could say it's one at each end of the replication bubble. It's the same thing. So as DNA is being unwound, like you can see from here at the top to here at the bottom, about 10 bases has been unwound or so on either side at least, maybe 20, 30, 50. For every 10.5 bases that gets unwound, one supercoil gets thrown into the DNA out here and has to be removed, or the DNA gets so kinked up that it won't, the helicase enzyme cannot open up any more DNA and replication would stop. Okay. Adding the next protein that's involved. So there's a lot of proteins involved in this process, but if you think through and learn the process, you know what needs to be done at each step, and there's the enzyme that does it. And that's the easiest way to remember this. The next enzyme is involved, or is usually hooked to or right next to the helicase and follows it. This enzyme is called primase. So in order for the DNA polymerase to synthesize DNA, we saw it needs template DNA, it needs a free 3' hydroxyl end at what's called a primer template junction. So let me go back to the So DNA polymerase, in order to add a nucleotide, needs template DNA, so it can decide, so that the DNA can decide what nucleotide to add next, and it needs a free 3' hydroxyl at a primer template junction. This is the primer template junction, where you go from double-stranded DNA to the single-stranded template DNA. So the DNA polymerase needs this, some double-stranded DNA with a free 3' end where the single-stranded starts. If you just gave the DNA polymerase this gray part over here and the sort of peach color part over here was gone, the DNA polymerase could do nothing because it needs this. It needs double-stranded DNA that ends in a primer template junction here in order to add nucleotides. Primase is, however, able to use a template DNA if this was not here. Primase can interact directly with a single strand of DNA that has no double-stranded primer hooked onto or base paired onto it. And so it's really important because on both the leading and lagging strands, if you didn't have primase, when the DNA opened up, you would just have two template strands. DNA polymerase would not be able to add any copied DNA and you would be done. All right? What's the catch? And there is one. Primase synthesizes RNA and not DNA. So if primase had this strand to work with, it could start way up here and start adding, just laying down nucleotides, but it would be a DNA strand on the template and an RNA strand here on the newly synthesized DNA. So the sugars would be different. They would have that 2' hydroxyl, and anywhere that there's an A, you would get a U instead of a T. So remember how earlier I talked about the hypothesis that RNA was the first nucleic acid and it served the functions of DNA proteins? Well, this is a holdover from that, we think. So the ability to just start synthesis with no primer on there was something that was only available to be done by RNA polymerases. And for some reason that just hasn't gone away. So at the origin of replication, pretend all this stuff right here and right here is gone, and all you have is the gray strand here and the gray, well, the helicase is there. You just have this gray strand and this gray strand. DNA polymerase cannot add a new copy to that. So you need primase to make the first strand. So 5' to 3' going left to right. On the bottom, 5' to 3' going right to left there. It only makes a short little piece of several nucleotides, and then it's done. But the job is finished then, because as soon as this primer is done or is made, primase lets go, and then because there is a primer here with a free 3' hydroxyl at the end of the RNA sequence, now DNA polymerase, even though this is an RNA DNA hybrid, so the template's DNA and the green here is RNA, DNA polymerase can come in here and use that 3' hydroxyl and start making this leading strand going right to left and this leading strand on the bottom going left to right. Notice that the lagging strand is not there yet because this is the first leading strand, and so you have to open up enough template here and here in order to even be able to make a lagging strand. Are we still together? All right. Stop me if you're confused. I mean, seriously. As soon as you get enough template over here and over here, then primase is going to lay down another RNA primer that's going to be the first lagging strand, Okazaki fragment. And then this RNA primer can be elongated from here to there by DNA polymerase, and from here to there by DNA polymerase. This is going to be this first Okazaki fragment. This one here is going to be elongated from there to there to make this first Okazaki fragment. And then you're moving apart. The leading strand here only needs one primer, because once it started, you always have a 3' hydroxyl at the end. You're good to go until replication ends. The lagging strand continually needs primase to synthesize RNA primers, because here there's the first one, it gets filled in, then when more sequence over here gets opened up, you need another primer to start the second Okazaki fragment. You need another RNA primer over here to start this second Okazaki fragment. Okay? And again and again and again and again. So the leading strand is going to have one RNA primer at the middle. The Okazaki fragments are going to have a whole bunch of RNA primers at the 5' end of each Okazaki fragment. And on you go. So here's the leading strand. Where I've put the little little symbols there is where the DNA polymerase would be working at the 3' end. DNA polymerase will start here at the 3' end of the primer and work down here. When you get to the point where there's no more nucleotides in the template, this is going to fall off. Same thing there. And then you will get more unwinding here, another primer being made by primase, and then the DNA polymerase is going to come in again. So the DNA polymerase we're talking about here is DNA polymerase 3. So the names I'm going to give you are the names of the polymerases used by E. coli. Eukaryotes have similar polymerases, but we're not going to worry about those. So don't worry about memorizing all these, because you don't have to know them, except for DNA polymerase 3, which we've already talked about, which elongates DNA, and DNA polymerase 1, which we're about to talk about, that's going to do a separate function. So all these other kinds of DNA polymerases are involved in, so 2, 4, and 5 are involved in various aspects of damage repair. So there are multiple pathways that if your DNA is damaged, they can go and fix those repairs. Yes? Yes. Question is the lagging strand primed several times? Yes. Okazaki fragments, I think, are about 200 base pairs, maybe a little bit more. It's similar length. Each species will have about the same length of Okazaki fragments, but I think that they differ between species a little bit. So every time you get a new Okazaki fragment, you have to have a new primer on the lagging strand. So the lagging strand will have hundreds or thousands of primers put on it, depending on how far it has to go. Whoops. Depending on how far it has to go before it hits another replication bubble. The lagging strand continuously gets new RNA primers every time you need to start a new Okazaki fragment, because the DNA polymerase can't start without it. But the lagging strand never has to start again, and that's why it only needs just one. Right, so primase works only one time on a particular lagging strand, but many times over and over again. Got it backwards. Primase works only one time on each leading strand, but many times on each lagging strand for a particular replication fork. All right, so at the end of DNA synthesis, the leading strand will be fine, but the lagging strand will have many Okazaki fragments, each of which has an RNA primer at its 5' end. And so while these were necessary to get DNA polymerase III able to add nucleotides to the end of the chain, we've got to get rid of them now. And so DNA polymerase I helps to do that, helps to affect that function. So DNA polymerase I has two functions that you need to know about. The first one is, as is shown down in blue there, a 5' to 3' exonuclease function. So what does exo mean? Outside, right? Insects have an exoskeleton. That means they have a hard covering outside of them. What's a nuclease? We already talked about that. With the DNase I nuclease sensitivity, right? And nuclease is an enzyme that is capable of breaking the phosphodiester bonds that hold nucleotides together. There are two kinds, an exonuclease and an endonuclease. This one is an exonuclease, which means it has to have a free end, and it will knock nucleotides off that end one at a time. This one has a 5' to 3' exonuclease activity, which means it can't look at the 3' end and remove nucleotides that way. It has to remove nucleotides 5' to 3', so this way. So the DNA polymerase I has a 5' exonuclease function, so it can chew off those RNA bases one at a time, breaks the phosphodiester bonds, and then each nucleotide, the hydrogen bonds won't be able to hold it on, and it falls off. It also has a DNA polymerase function, 5' to 3' polymerase. So as it's removing one after the other nucleotides here, it's coming behind and elongating the chain. So this is an easier way to see what's going on. So this is the 3' end of the other Okazaki fragment. So if you think of this end here, there's going to be, where I'm drawing with the pointer, another primer. So this is going to be happening here as well. So the 5' to 3' exonuclease activity is removing these RNA bases shown in green by knocking them off one at a time, and away they go. The DNA polymerase function is coming behind that and adding new bases to the chain. So it's removing the primer and adding DNA bases to replace it. So we're replacing the RNA bases in the primer with DNA bases, which is what we want to do. Problem is, you're left with a nick at the end, right? And a nick is just a single-stranded DNA break. So this strand that was the template has a solid sugar phosphate backbone all the way across. But when the last one, let's pretend these are DNA bases now, and we're to the end. So you could add the next one, you'd add a T here, and then you'd have, this is the end of one Okazaki fragment, here's the beginning of the other one, but there's no phosphodiester bond between there, because the polymerase can only make phosphodiester bonds, as I said earlier, by adding nucleotides, and there's not a nucleotide to add. So you have, here you have a 3' hydroxyl and a 5' phosphate, but the polymerase just falls off and can't fix it, right? In comes the last enzyme that's going to work in this pathway, or this function. It's called DNA ligase. DNA ligase is able to do just what I said. It can take a 3' hydroxyl and a 5' phosphate, where you have one strand with a nick in it and the other strand solid, and it can join them. So it uses the energy, of ATP in the cell to actually do this. So DNA ligase forms the phosphodiester bond that is missing, and then that's it. So DNA polymerase 1 will use ' 5' to 3' exonuclease activity to remove the RNA bases from the primer. It will use its 5' to 3' DNA polymerase activity to replace those bases with RNA bases with DNA, and then it leaves a nick, which is ligated or brought together by DNA ligase enzyme, and that's it. Now you have 2 strands of DNA. The leading strand, which is not shown here, doesn't have to have any of this happen to it except at the very beginning, because there's only one RNA primer involved. The lagging strand has to have this mechanism happen over and over again. But now it's done. So here is the big bad table with all the proteins that you need to know. And you need to know what they do. But the way to learn this is not to sit this table sit this table on your table and memorize it. It's to have this table out while you're reviewing the entire process we just talked about. You know how I ask questions and stuff now. I can't ask you what each one does, but you have to say in your own words what each one does, and I will ask other questions that, where does it work and, how does it affect its job? So the best way to study this is to review the entire process and how it works. Just go through it a bunch of times with this table next to you. And pretty soon you'll have it. All right, so let's go through and look at this all together now. So here is a replication fork. Remember, one fork at each side of a bubble. This is the fork going from left to right. There's going to be another fork over here going right to left, and everything that happens on this one is going to be just the opposite on the other one. So the helicase is up here, and it's not shown, but primase is always hanging out with helicase and rides along with it. Helicase is moving from left to right, and it's breaking it's using ATP to break the hydrogen bonds that hold the DNA together. Right? You have DNA gyrase that's going out in front of the replication fork and allowing the DNA to unwind. The gyrase is not unwinding the DNA and breaking the hydrogen bonds, but it is removing the supercoiling that helicase generates. Then every so often the primase hooked up to helicase adds a primer onto the lagging strand. The leading strand is going from left to right here. The 2 tan circles represent the active sites of the DNA polymerase. As we'll see coming up, the DNA polymerase is a 3-headed monster, just like the dog in Harry Potter, right? There's one of the 3 active units that moves on the leading strand and always stays on there. As we'll see, the other 2 active polymerase subunits are going to be tag teaming to take care of the lagging strand. So there was kind of a weird, we don't understand how this works going on in the field up until maybe like 7 or 8 years ago, where scientists know that in real life, DNA replication takes place as fast as you can do just one of these polymerase subunits going. So you can separate each of these polymerase subunits in a test tube and just throw it in with some DNA and whoosh, there it goes, right? The whole process was occurring as fast as you could just do the leading strand by itself in a test tube. And scientists were wondering, well, how can that be? Because on the lagging strand, so you're moving, the polymerase is moving from here down to here, and then there is a slight time lag where it has to let go here, find the next primer, and start again. But there wasn't any time for that because it could go, the whole polymerization could go as fast as the leading strand was going. That led them to discover that they thought that the DNA polymerase had only 2 heads and now it has 3, because the other 2 are tag teaming. While this one is polymerizing here, the other one, the third one, is moving up to find this primer, so that by the time this polymerase is done here, the other one is already up there and can start, and then this one will let go and move up to the next primer. Did that make sense? Okay. So now we're finishing this, moving up there, and the 4th primer is almost ready to go. So now the second primer is already done. This is the other subunit. So this polymerase is going to let go and be going up to look there, while the other one has already found this one. This is the second one working on the 3rd primer here, and the other one is probably already up here getting ready to go. So on the leading strand, it's one polymerase just going zoom down to the end. On the other strand, it's like, I'll do one and you'll do one, and I'll do one and you'll do one, right? Over and over again with those other 2. All right. So in eukaryotes, we also have the problem of removing and adding histones to make nucleosomes. So the nucleosomes have to come out of the DNA to have replication occur because the DNA is wrapped around them. So nucleosomes are also duplicated during DNA replication because at the end you're going to have twice as much DNA, so you're going to need twice as much of the histone H2A, H2B, H3, and H4 proteins, and also H1. So here are 1, 2, you can see the little opening there. Here's a little one, here's another one. These are replication bubbles. This one's bigger. But you can see, here's the original DNA, here's where it's opened up, and you can see as soon as, here's a smaller one, as soon as the new DNA is synthesized, you can already see that it's wrapped around the histones there, like the little beads on a string again. So it's pretty quick. The histones come out, DNA is replicated, and it wraps right back up around the histones. All right. And so the question became, how does this work? So is it the old histones stay together in that octamer and then you make new octamers and they go in, or how is this done? Similar to the DNA replication experiment, instead of using DNA that was labeled with heavy nitrogen, remember N15N, now you're growing cells in heavy amino acids for many generations, so that all the amino acids that are in the nucleosomes are heavy. Then you take out the octamers and look, you get a band at the heavy area. This is the same kind of gradient centrifugation as was done in the Michelson and Stahl experiment. Then change media, go to light amino acids, and allow one round of replication to occur. And remember, in the Michelson and Stahl experiment, after one generation you got a band right in the middle, and it was a very tight band, so not very spread out. And that was because each strand had exactly half heavy and half light nitrogen. Here you get more of a diffuse band because the old and new octamers kind of mix together and reform. And after 2 generations, you don't get a light band and the band in the middle. It's just, it becomes more and more diffuse and moves up toward the top. So exactly what would happen if dispersive replication would happen for the DNA. And so what actually happens with histones is that the subunits, when they come out of the DNA, dissociate into H3H4 tetramers and H2A, H2B dimers, and those never come apart. So the tetramer, the H3H4, is either all old or all new, and each H2A, H2B dimer is either all old or all new. And then they're all of those tetramers and dimers are just floating around in the cell and randomly go back into the new, form the new nucleosomes. So a new nucleosome could be all old, heavy histones, all new light histones, or any mixture. Like the H3H4 could be old or new tetramer, all one piece, and each of the two H2A and H2B could be old or new in any combination. That's why you get this diffuse band here, showing that it just gets scrambled and mixed up. Good? All right, so now the coup de grace. So, sort of. So we got to finish this up. If you're doing circular replication, everything is fine. So this is showing just one of the two DNA molecules. So half of this, so if you start this way going that way, this would have been the leading strand down to here. This would have been the lagging strand here. But we're all finished with that process, so all of the RNA primers have been removed from this, and you have like the one final primer here that's ready to be removed. So the RNA primer could be removed by DNA polymerase 1, filled in by DNA polymerase 1, DNA ligase seals the nick by making a new phosphodiester bond, and that's it. You would have this ring catenated or like around another ring, and you would just need the topoisomerase 2 to break one of the rings and then have them go back together, and you're done. Because it's circular, we're going to run into a problem with linear DNA and we're coming back to talking about telomeres again. So remember, we talked about in the last chapter how they shorten. We're going to find out why that happens and what the cell can do about it. So here we go. Here's the leading strand. This is the lagging strand, leading, lagging, depending on where you're at in the replication bubble. Most of the RNA primers, because they're internal here, are removed, filled in by DNA polymerase 1. Nick gets sealed by DNA ligase. Everything is fine until you get to the very end, the last lagging strand at the very end of the chromosome, and you're going to be in trouble. The reason is because you always get this 3' overhang here. This last primer, imagine if it's on the end here, so if this was all primer, you can have, this can be removed by the 5' to 3' exonuclease activity, but you can't have DNA added here with the template, because what does the DNA polymerase need? And even the DNA polymerase 1's polymerase needs a free 3' hydroxyl, and there isn't one over here. So you're stuck. You get this gap left at the end. So no big deal, because you need a 3' end to tuck in to the sequence like I showed you in the last chapter. Except it becomes a big deal when this strand right here, which is already too short, is used as a template in the next round of replication. So when this strand is used as a template, pretend this gray strand is not there. This is going to be the leading strand. Here it comes. The polymerase is coming down to the end. And boom, it stops right here. This part that's over to the right never gets made. Why? Because the trains run off the track. There's no more template here. See how that works? And so now the DNA is shortened by that much. And then when this strand that's already shortened to here is used as a template next time for the lagging strand, pretend this isn't here, now there's going to be about this much more, not put it, the same thing for the RNA again, and then when it's, then pretend all that's not there. When this is used as the leading strand again, synthesis stops here. And so you can see it's not a big deal now, but each generation, that 3' end gets shorter and shorter and shorter and shorter. And after about 50 generations, enough is gone that the cell senses too much telomere is gone and stops dividing. It's bad news if you're in a situation where continuous mitosis is necessary. So enter the telomerase enzyme. So telomerase, like the ribosome and like the spliceosomes that are going to splice out introns that we talk about later, has both protein and RNA components in it. So in this case, the proteins, and there's multiple proteins that go together, are shown by the yellow sequence there, and it also has on board this little green RNA molecule. The RNA molecule is going to match the telomere sequence. Remember when I talked about telomeres, they're all a 6-base pair repeat. In this case, it's TTG, GGG. See? TTG, GGG, TTG, GGG, etc. over and over again. The RNA sequence is the complement or the template for making more telomere sequence. And so the part of the protein here is a reverse transcriptase. So remember with the Watson and Crick central dogma, they said DNA is transcribed to RNA, translated to protein, and then somebody discovered that in retroviruses you have this reverse transcriptase activity that can make RNA back into DNA. Like, well, that's just for these viruses. We don't do that. And then somebody else, 3 people discovered actually that you do have reverse transcriptase that's required in our cells as well, and that's how you keep your telomeres long. So the reverse transcriptase uses the RNA template to make new DNA sequence. So it's a polymerase that uses an RNA template and makes a DNA copy of it, like here. So in addition to the reverse transcriptase activity, telomerase also has a helicase activity. So after this synthesis is done, the helicase breaks the bonds that are holding the template onto the newly synthesized DNA, and it moves down 6 bases. And then the process repeats. Add more DNA with the reverse transcriptase activity, then the helicase activity will cause this to be released. It moves down again, and repeat and rinse and repeat, right? until the telomere is long enough, at which point the telomerase falls off and another Okazaki fragment is added here, which, it's going to be longer than this. This is just for demonstration purposes. And you'll always have that free 3' end, but it doesn't matter. That's necessary to tuck in, and the telomere is long enough, and everybody wins. Well, how does this telomerase enzyme know that the telomere is long enough? We don't go into specifics in this class, but there are actually telomere binding proteins, that a complex that binds to the end of the telomere. In addition to protecting the end of the telomere helping it tuck in there like we showed in the last chapter. Each one of these complexes also has a very small but very real inhibitory effect on telomerase. And so when you get enough telomere binding proteins, the combined inhibitory effect of all of those is enough that it's like, okay, telomerase, time to go, right? And then telomerase doesn't come and work on that end until it's shortened up after enough generations that there's few enough telomere binding proteins that it can come in and not be inhibited. So it's a homeostasis mechanism. See all the stuff you talked to homeostasis, right? When are we ever going to use that? Right here and right now. The homeostasis mechanism. So this same mechanism we'll see used when we talk about adding the poly-A tail. Does anybody remember that from way back when, right? Each eukaryotic messenger RNA is going to get a bunch of A's added to the end of it. The enzyme, the poly-A polymerase that makes that tail, is going to keep doing it until it's inhibited by a large enough concentration of poly-A binding proteins, just like this. All right, we are close enough to the end that I will end there. So here's the deal. The exam is on Tuesday. You will need a Scantron. You will need, you know, whatever. You can have 2 pieces of scratch paper again if you want, all that stuff. I don't know that you'll need them. Fine. On Thursday, I'm willing to use some of the class time for a review. The review part of the class will last as long as you guys keep asking questions. Right? When you guys run out of questions, specific questions to ask that shows you've been working on the material and want to know this and this and this about it, then I'll go on into chapter 13. So I'll put up the PowerPoint. Please read ahead a little bit into chapter 13. The exam will only cover to where we are at right now, the end of chapter 12. So anything we talk about on Thursday, chapter 13, will not be on the exam. Good? All right, so I'll see you guys in the lab today. Remember, if you're in the lab today, there's going to be a 45 to 50 minute downtime, with which you'll be able to work on homework or leave as long as you come back, whatever. So please bring something to do. Also, remember the BAT talk is tonight, not here, it's in the library tower. 10th floor, we'll have little chicken salad sandwiches, not from the chicken salad chick, it'll be the Costco chick instead. but we'll have some of that. Dr. Grillo is going to bring a number of preserved bat specimens to look at, and I have some bat cookies that came from our kitchen. If you show up, you need to sign up. I will have an attendance sheet that I will put out at the end when it is over, and you will get 5 points for this class if you show up and sign the thing at the end. See you then.